mitochondrial disease. Okay. This topic will be approached as what is mitochondria, what is cytoplasmic inheritance, what is oxygen transport chain, then causation of disease in this group, and classification of common neurological diseases of this group, then bedside clues and take-home message. Ancestry, it is said that mitochondria arose through a fateful endosymbiosis more than several billion years ago. And mitochondria descended from a specialized bacteria. This is a non-sulfur bacteria which got incorporated into the living cell by endocytosis by a species of prokaryote or some other cell type, and then became incorporated into the cytoplasm. So actually it is another living thing which got incorporated into the human cell. Not any living cell several years ago, and then it became the energy machinery of the cell. And human mitochondria evolved from an alpha proteobacterium with an archaeal derived host cell. It's an algae that was mostly related to Asgard archaea. And they can be thought as batteries or energy factories of the cell. So what happened is another living organism got into another living organism via endocytosis, and that became converted into the energy machinery and for that living cell. In the human beings also, same process happened, and this got incorporated through an algae, and then it became the battery of the human cell. So what is this mitochondrial genome? So mitochondrial genome is a small circular DNA. We know that nuclear DNA is not circular, and this is a circular DNA, and it encodes 13 proteins of the OSPHOS system and 22 mitochondrial tRNA and 2 mitochondrial rRNA. So this constitutes the mitochondrial genome. It's a ring shaped molecule, and unlike the nuclear DNA, which is always seen in the nucleus, it is seen in the cytoplasm, and it codes for 13 proteins of the OXPHOS machinery, that means oxidative phosphorylation machinery, and 22 mitochondrial tRNA and 2 mitochondrial rRNAs. Several thousand copies are made per cell, unlike the usual 23 in haploid and 46 in diploid. We know that the nuclear DNA is 23 pairs in the reduction division, 23 in the reduction division, and 46 in the mitosis, whereas this is several thousand copies of the mitochondrial DNA are there in the cell. 90% of the body energy is produced by the mitochondria, and it is saved as ATP. Here we can see that this is the nucleus. This is the nucleus, and this is the cytoplasm. And in the cytoplasm, we got, this is the only location where the genetic material is situated in the cytoplasm. So in the cytoplasm, the mitochondria is situated. And this is an amplified view showing the nucleus and several mitochondria there that has an outer membrane and inner membrane and intercellular space that I will be expanding later. So most important to remember that it is the only situation where the genetic material is seen in the cytoplasm. So now in 1963, researchers discovered that mitochondria have their own DNA or blueprint mitochondrial DNA, which is different than the nuclear DNA found in the cell's nucleus. So everywhere DNA is present only in the nucleus. This is the only situation where it is in the cytoplasm. And mitochondria have their own DNA consisting of a double-stranded circular molecule. This mitochondrial DNA consists of 16,569 base pairs that constitutes 37 genes. And here we can see what is the chloroplast. So in the plants, the energy producing structure is the chloroplast. And this is akin to, mitochondria is akin to the chloroplast in the plants. And it supplies energy to eukaryotic cells, and it mostly requires oxygen and contains their own DNA. And here we can see that it has got a folded membrane within a outer membrane. There's an outer membrane and there's a folded membrane. This folded membrane is called cristae, and function is converts energy stored in food materials into usable energy. The energy stored in food materials is not in the usable form. Here it becomes converted into usable form, that is ATP, and allows aerobic cellular respiration. Here you can see the outer membrane, the inner membrane, the cristae, the circular DNA inside. Then you have got the phospholipid granules, peroxisomes. Each one produce a different group of disorders. And you have got the inner membrane where there is ion channels are going. So grossly, the functions are outer membrane has lot of transport channels. The inner membrane is involved in electron transport. And the inner membrane, that's the space between the outer smooth membrane and the inner folded membrane, has electrochemical gradient. And matrix enzymes form oxidative phosphorylation system. And what is this genes in complexes? Oxygen transport chain consists of 5 complexes. Complex 1 is situated in the nuclei, and the genes involved are many of them, and we can produce disease. And complex 2 is again nuclear, mitochondrial nuclear. Complex 3 is mitochondrial nuclear. Complex 4 is also mitochondrial nucleus. Complex 5 is cytoplasmic. And so these are the 5 complexes in the oxygen transport chain. So what are the peculiarities of this genetics? It is a uniparental inheritance, maternal inheritance. This comes from the mother. And why it comes from the mother? Because when the zygote is formed, only the cytoplasm of the mother is used to form the zygote. Paternal zygote is discarded. And as this structure is present in the cytoplasm only mother can transmit the disease. And high copy number. We saw that in the nuclear DNA, there is only 23 pairs, where here it is a high copy number. Heteroplasmy, that means there will be a mixed type of genetic material, both carrying disease and both normal in huge number. When there is a particular number of abnormal ones, disease manifests. Few number of abnormal one does not cause disease. That is called heteroplasmy. So there will be a different kind of disease and normal genetic material, and if the load is more than 70% only, disease becomes manifest. And what is bottleneck and segregation? Bottleneck effect is one patient might get the maximum number up to 70% of abnormal mitochondrial genes, so he becomes symptomatic. Another child in the same family have only 10%, he may transmit, but he doesn't become symptomatic. That is called bottleneck effect. And threshold effect is the amount of defective mitochondrial DNA needed to cause disease. That is about 70%. And next is dual genetic control. What is that? Mitochondria contains the genetic material in the cytoplasm, but this mitochondrial DNA can be controlled by the nuclear DNA. So there is a dual control. So it can be purely a mitochondrial DNA disease or a disease of the nuclear DNA's control over the mitochondrial DNA. In that situation, the inheritance need not be maternal. It will look like a autosomal recessive, autosomal dominant, or X-linked, and the commonest being autosomal recessive. That's a nuclear coded mitochondrial DNA disorders and purely cytoplasmic mitochondrial DNA disorders. The magnitude of the problem is one of the commonest inborn error of metabolism, and its prevalence is 1 per 5,000 live births, and one child is born every 30 minutes who will develop mitochondrial disease. So 1 in 200 population have mitochondrial mutation that may lead to disease. I said about the bottleneck effect. So 1 out of 200% have the mitochondrial mutation, but until the bottleneck effect manifests, they will not get the disease. And it can affect the central nervous system, cardiac muscle, kidney, skeletal muscle. And generally, each person will have involvement of more than 3 or more organs without a unifying diagnosis. Each organ will not have a unique diagnosis like a heart attack. Like that will not be there. They will have cardiomyopathy. So why this happens? So that kind of a non-unifying disease of the multiple organs. And these have been categorized into class 1 and class 2 oxidative phosphorylation defect. Class 1 disorders result from mutation of mitochondrial DNA itself. We saw that the mitochondrial DNA is in the cytoplasm. So mutation taking place in the cytoplasmic mitochondrial DNA itself is called class 1. That is the uniparental inheritance, comes only from the mother. Class 2 is disorder of the cytoplasmic mitochondria due to defect in the control of the nuclear DNA over the cytoplasmic DNA. So what happens? It will be autosomal dominant, recessive or X-linked. That comes as class 2 disorder. So how do we classify? Apart from the pattern of inheritance as class 1 and class 2, clinically, we can classify them based on the clinical features, biochemical defect and genetic mutation. Either way, in each 3 different ways, we can classify them. So based on the clinical features, we have got both syndromic disorders and non-syndromic disorders. Syndromic Kishan Rao syndrome. I will show classical cases of each one. Melas, that is mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. Murph, that is myoclonic epilepsy with ragged red fibers. Leon, that is Leber's hereditary optic atrophy. And chronic external ophthalmoplegia. MNGIE, mitochondrial neurogastrointestinal encephalopathy. Lees disease, that is encephalopathy of the young child. Alpers disease, where there is partial anencephaly, corpus callosum defect, abnormal ear development, and NARP, that is neurogenic weakness with ataxia and retinitis pigmentosa. These are named syndromes. Non-syndromic mitochondrial disease can present as extrapyramidal disorder, neuroregression, ataxic syndromes, progressive myoclonic epilepsy, epileptic encephalopathy, limb-girdle syndrome, peripheral neuropathy, episodic encephalopathy, and ataxia with neuropathy. That is clinical classification. Then classified based on biochemical defect. So we thought there are 5 complexes. So it can be complex 1, complex 2, complex 3, complex 4, complex 5, or multiple complex deficiency. So that is based on the biochemical defect. Then based on the genetic mutation, mitochondrial DNA mutation may be point mutation of mitochondrial DNA, single deletion duplication of mitochondrial DNA, or it may be nuclear DNA mutation of genes related to part of the enzyme, genes related to transportation of mitochondria, mutation of genes related to mitochondrial biogenesis, and mutation of genes related to functions of mitochondrial DNA, that multiple deficiency, duplication, and defect of multiple mitochondrial DNA. So this is after the genetic testing. So we can classify based on clinical features, we can classify based on biochemical defect, and we can classify based on the genetic defect. And classification, rearrangement, that is deletion, duplication. We have got these conditions. Very common, chronic progressive external ophthalmoplegia. Kishan Rao syndrome is not uncommon, but it is less common. And we have got the single nucleotide variants, Leber's hereditary optic atrophy, NARP, Lees syndrome. Then T-RNA, we saw that there is 22 tRNA and 2 rRNA in the nuclear mitochondria, in the cytoplasmic mitochondria. And this tRNA produces Melas, Murph, progressive external ophthalmoplegia, myopathy, and cardiomyopathy, diabetes, deafness, and non-syndromic sensory neural deafness. And we have got rRNA, that is aminoglycoside-induced non-syndromic deafness. The nuclear genetic disorders of the mitochondrial respiratory disorder is mutated genes encoding structural subunits. Again, Lees syndrome, optic atrophy. Mutated genes encoding assembly factors. That is Lees syndrome, leukodystrophy, tubulopathy. And mutated genes encoding translation factors, Lees syndrome. Lees syndrome is seen in, it's a polygenic condition. And you have got associated multiple mitochondrial DNA, progressive external ophthalmoplegia, Mingy, and Alpers, and ataxic neuropathy, and others will be coenzyme Q10 deficiency. Then what is the surprise? We saw that 90% of the oxygen carriage, ATP production is done by the mitochondria. And mitochondria is the site of cellular respiration and powerhouse cells. And it converts glucose to ATP. But strangely, RBC, a major oxygen carrier, has no mitochondria. So RBC is the only organelle in the human body which does not have mitochondria. Everywhere mitochondria is there. RBC, a major oxygen carrier, is not there in the oxygen transporting mitochondria. Oxygen transporting mitochondria is not there in the RBC. The function of RBC is to carry oxygen using protein hemoglobin. So function of RBC is not transporting oxygen through oxygen transport chain. It is through hemoglobin. And to accommodate large quantity of hemoglobin, the RBC is enucleated and mitochondria is absent. And cellular respiration. So that is a surprise. Another oxygen transporter does not have mitochondria. And where does cellular respiration take place? It actually takes place in two parts of the cell. Glycolysis occurs in the cytoplasm, and Krebs cycle and electron transport chain occurs in the mitochondria. So mitochondria are organelles found inside most eukaryotic cells. They generate the energy that the cells need, so they are sometimes compared to power plants. So it's like the power plant. The mitochondria makes adenosine triphosphate, which the cell can use as an energy source. The synthesis of ATP is done through the citric acid cycle, which uses glucose, pyruvate, and NADH as the input molecule. And production of the energy currency of the cell, that is ATP, and this happens by the citric acid cycle or the Krebs cycle, and generates the high energy electron carrier NADH and FADH2 through the oxidation of acetyl coenzyme A to carbon dioxide coupled with the reduction of NAD and FAD. 5 protein complexes referred to as complex 1, 2, 3, 4, 5 embedded in a tightly folded inner mitochondrial membrane. Complex 1 to 4 constitutes the electron transport chain and move the electrons down the assembly line. Complex 5 makes ATP and it is called ATP synthase. Complex 5 is called ATP synthase. Apart from oxygen transport, it is involved in calcium homeostasis, regulation of innate immunity, programmed cell death, and stem cell regulation. In the electron transport chain, electrons are passed from one molecule to another, and energy released in these electrons are இந்த படத்தை ஒன்று மாத்திரம் இந்த இது இவ்வளவு இதுதான் சைடில். So in the electron transport chain, electrons are passed from one molecule to another, and the energy released in these electron transfers used to form an electrochemical gradient. Oxygen sits at the end of the electron transport chain, where it accepts electron and picks up protons to form water. Oxygen acts as a final electron acceptor that helps move electrons down a chain that results in adenosine triphosphate production. So electrons are transported through complex 1, 2, 3, 4, and fifth complex accepts oxygen, and water is formed and ATP is also formed. So what are all the complexes in the electron transport chain? So oxidative phosphorylation. Oxidation means oxygen transport. Phosphorylation means formation of ATP. So oxidative phosphorylation is the metabolic pathway in which electrons are transferred from electron donors to electron acceptors in the redox reaction. This series of reactions release energy which is used to form ATP. There are 4 protein complexes labeled 1 to 4 we saw, and in the electron transport chain, which are involved in moving electrons from NADH to FADH to molecular oxygen. Complex 1 establishes the hydrogen ion gradient by pumping 4 hydrogen ions across the membrane from the matrix into the intermembrane space. We saw the outer membrane, intermembrane space, and the inner membrane. So complex 1 establishes the hydrogen ion gradient by pumping 4 hydrogen ions across the membrane from the matrix into the intermembrane space, that is between the outer membrane and the inner membrane. Complex 2 receives the FADH2, which bypasses the complex 1 and delivers electron directly to the electron transport chain. Then ubiquinone accepts the electron from both complex 1 and 2 and delivers them to complex 3. Then you have got complex 3, pumps protons through the membrane and passes its electron to cytochrome C for transport to the 4th complex of the protein and enzymes. Complex 4 reduces oxygen. The reduced oxygen then picks up 2 hydrogen ions from the surrounding medium and makes water. And so what is the cytoplasmic inheritance? I have already mentioned this. The mitochondria, so we saw just grossly how the mitochondrial functions. The mitochondria and thus the mitochondrial DNA are passed exclusively from the mother to offspring through the egg cell. Children inherit this mitochondrial DNA with this much of functions only from their mother, because when the sperm fuses with the egg during conception, the sperm's mitochondria is destroyed. So we saw it is a double-stranded one, and the only place where genetic material is outside the usual chromosome, inherited through mother. It has 16 kilobase length and 16,000 base pairs and codes 37 genes and 13 proteins that are subunits of oxidative phosphorylation, and it has only exons. It is very important. Therefore, there is no repair. So introns are needed for repair of genetic material, whereas in mitochondria, only exons are there. Therefore, there is no repair. Once damage comes, the damage remains. And so no repair mechanism and close to oxidative stress, and hence lot of mutations. Because of that, only the 1 out of 5,000 children born have mitochondrial disease, because there is no repair mechanisms. So the sick mitochondria remain sick. And there is homoplasty, heteroplasmy, and segregation. What is homoplasty? One cell may have all diseased mitochondria, and one cell may have all normal mitochondria, and or varying degree of segregation, the bottleneck effect. So that within one family, there may be several children with several mitochondrial diseases, but they may not be uniform. Like if you see 4 brothers having epilepsy, that is a uniform disease. Here one person will have Kearns-Sayre syndrome, another person will have Lee's disease, another person will have mitochondrial cytopathy, like that. So because it depends on the number of mutant mitochondria received by each cell. Homoplasty, they will be normal or totally abnormal, and they may not be compatible with life. Heteroplasty, the varying degrees of abnormal mitochondria decides the phenotype, and segregation to one group of diseases results in the classical phenotype. That is the bottleneck effect. And mutations are more due to lack of protective histones and absence of introns. And there is oxidative stress. And as I said, heteroplasmy and homoplasmy, that results in a spectrum of abnormalities in the children in the same family. And threshold effect, the frequency of mutant has to reach a desired number, hence phenotype changes. So depending on the number of mutant mitochondria, the phenotype will be different. And mutations in the genes encoding the mitochondrial DNA maintenance machinery can lead to either mitochondrial depletion or multiple mitochondrial deletions. And that is when the maintenance is defective. There are mitochondrial DNA maintenance machinery. So that maintenance machinery is sick, means there can be depletions of a single or multiple DNA particles. And during meiosis, that is a reduction division, there is no recombination. We know that in the nuclear DNA material, during reduction division, the genetic material crosses over. That is called crossing over. And in the mitochondrial DNA, there is no crossing over. And the disease is a heterogeneous group of disorders as a result of dysfunction of the mitochondrial respiratory chain and mutation of genes, either nuclear DNA or mitochondrial DNA. So it can be, as we saw, class 1 is purely mitochondrial DNA, class 2 is nuclear coded mitochondrial DNA disorder. And mutations affect varying proportion of the many mitochondrial DNA molecules, that is heteroplasmy. So it can be a single organ disease or it can be a multiple organ disease. So it is uniparental maternal transmission. So maternal transmission happens in X-linked diseases also. So how do you differentiate the maternally transmitted X-linked disease from maternally transmitted mitochondrial disease? X-linked disease, mother is transmitting because the boys have X and Y. Mother is X and X. So when you have two X, the X-linked diseases do not manifest. Whereas XY, it is almost like 1X only. So the disease is transmitted by the mother and experienced by the sons. And usually mother is asymptomatic. Male children are affected as they have only 1X. Whereas maternal transmission is there for mitochondrial disease also. But the mother transmits not through the X. She transmits through the cytoplasm. So both males and females become affected. And mother is also symptomatic. And both gender is affected. That is how mitochondrial disease differs. The common thing is maternal transmission. Both are maternal transmission, but both boys and girls can be affected, and mother herself suffers from the disease. So this is the bottleneck effect, showing that these are one type of mitochondria, these are another type of mitochondria. See the proportion of different mitochondria, normal and the abnormal, depending on the varying types of phenotypes will happen. And this restriction of amplification event can lead to a random shift of the mitochondrial DNA mutation load between generations and is responsible for the variable level of mutated mitochondrial DNA observed in the affected offspring from mother with pathogenic mitochondrial mutations, because they are randomly distributed. Unlike the nuclear DNA, which has got a pattern, mitochondrial DNA has no pattern. They are randomly distributed, huge number, varying degree of combinations and segregation. And this physical bottleneck is a physical reduction in the copy number of the mitochondrial molecules per cell, which occurs during development. As described below, the genetic bottleneck or the mutant proportion spread. We saw that each cell has got different number of mutant proportion. Only if it is 70%, disease comes. So may vary from species. So who decides this bottleneck? So many people will be asymptomatically having mutant mitochondria, but if the number is less than 30%, they will not suffer from the disease. So the variation depends on the species, the individual's innate characters, the duration taken for the genetic material to get transferred, and mitochondrial DNA sequence and other factors. This causes subclinical disease. That means they may have mild fatigue or mild air hunger, or somebody may be having a fatal disease. So varying degrees of severity happens like that. So what is this mitochondrial homeostasis? We see that when there is a mitochondria, there has to be a homeostasis. It has got a life like any other cell. So what is this homeostasis? Elimination of damaged and superficial organelles. So after a particular period, when the life of the mitochondria is over, it should be eliminated. If it is not eliminated, it stores as toxic material in the cell and causes disease. So that can be homeostasis disorder, where there can be problem in elimination of damaged and superfluous organelles. Second is generation of newly synthesized mitochondria. So that has to normally happen. Once the abnormal mitochondria is expelled, new mitochondria should come. So there can be a defect in the synthesis of new mitochondria. The mitochondrial and cellular homeostasis may not be preserved, resulting in a group of disorders. And cellular adaptation and survival under stress condition may be poor. So under stress, patients' mitochondria become sick and produce symptoms which is not normally there, but under stress it becomes symptomatic. So these are the normal homeostatic mechanism, and each one of them can fail, resulting in a group of mitochondrial disorders. So this is runaway mitophagia, where the abnormal thing accumulates and a new synthesis is defective, and preservation of homeostasis is defective, and there is ROS means reactive oxygen species generation instead of normal oxygen transport, resulting in oxidative cell death. So next is homoplasmy and heteroplasmy. We have already discussed that. Only a pure population of normal. So in a family, 4 children, one may be perfectly normal or one may be fully abnormal and die at birth. The daughter cell may receive a mixture of mitochondria, some with and some without disease, that is heteroplasmy. So one having all normal, and one having few number of normal and abnormal, one having more number of normal and abnormal, that is heteroplasmy. So 1 out of 5,000 live births, generally bad obstetric history. So because of the bottleneck effect, one child may be born with 100% sick mitochondria. So they may present as multiple abortions. So that is bad obstetric history. If the mother has got a bad obstetric history, we have to suspect there may be a mitochondrial disease and there is natural selection. Nature eliminates the weak. So abortion is nature's attempt to eliminate the weak. So multiple abortions means they may have a mitochondrial disease. You have to suspect that. And phenotypic variations in the same family. As I said, due to the heteroplasmy or the bottleneck effect, each one depending on the type of abnormal mitochondrial inherited, they will have different types of mitochondrial disease. And common features are short stature, deafness, eye movement problem, lipomas, excessive hair, blindness, multi-organ failure, neuropathy, cardiomyopathy, diabetes, renal tubular acidosis. And mutations involve both mitochondrial and nuclear DNA. We saw that class 1 is mitochondrial DNA, class 2 is nuclear DNA. So nuclear follows Mendelian pattern. That is dominant, inheritance, and X-linked. And mitochondrial DNA acts as a slave of the nuclear DNA. Even though mitochondrial DNA is generally independent, the nuclear DNA has got a control over the mitochondrial DNA. So in class 2 conditions, the slave effect becomes altered in different patterns. So variability in phenotype due to heteroplasmy number based on bottleneck effect, role of nuclear DNA on mitochondrial DNA, and expressivity factors, all these cause disease. And we saw also the defective mitochondrial homeostatic mechanism. So this is the two types. One is the class 1, second is the class 2, where nuclear mutation and mitochondrial mutations. So some mitochondrial myopathies that may be subject to the rules of maternal inheritance, that is coming only from the cytoplasm, or MRF, mitochondrial encephalopathy, ragged red fiber syndrome. Melas, mitochondrial encephalopathy, lactic acidosis, stroke-like episodes. NARP, neuropathy, ataxia, retinitis pigmentosa. PUR, progressive external ophthalmoplegia. This is mitochondrial intermittent ataxia, leukoencephalopathy. A nuclear inheritance usually follows autosomal recessive pattern and rarely dominant or X-linked. Sometimes it may be sporadic. And disease severity varies depending on the ATP needs of the tissue. If a tissue which needs more ATP like brain or eye or ear, they suffer more. And tissues which need less ATP suffer less. And we have got a criteria. One is, so many criteria are there. I will be mentioning some of them at the end also. Modified Walker criteria, you should have 2 major or 1 major with 2 minor. So the criteria are categorized under clinical, histological, enzymological, functional, and molecular and metabolic factors. So all these parameters are used to form the Modified Walker criteria. And this you can, all of you can get it from the net. This is the Modified Walker criteria applied to children with mitochondrial disease. So 2 major or 1 major with 2 minor qualifies for the mitochondrial disease. And it can affect brain, strokes, seizures, mental abnormality, blindness, and hormone deficiencies, liver disease, and GIT disorders, muscle disease, deafness, cardiomyopathy, diabetes, renal disease, anemia, and neuropathy. So this is one of our child. I told about Lee's disease. That's the fatal encephalopathy, fatal encephalopathic form. So he's a 1-year-old boy. He presented with hypotonia, dystonia, deafness. He was a flaccid, he is a floppy child, but you can see the abnormal posturing of the limbs, and he is deaf, and his urine showed mild methylmalonic aciduria. And you can see the classical MRI appearance showing symmetrical hyperintensities in the putamen. And this is mitochondrial DNA depletion syndrome. It is fatal. Usually these children do not survive. So this is deficiency of multiple respiratory chain in the complex. Complex 1, 2, 3, 4, 5 visa. Multiple complexes are defective. They present with severe cognitive and developmental delay with deafness and dystonia. It's not playing. Okay. So next is the muscle disease. So that is the least disease. Second, the muscle diseases can present with other organ involvement also. So patient with myopathy, you can think of LGMD or Duchenne, so many muscle diseases we'll consider. But when a patient with myopathy has involvement of other parts of the neuroaxis, other part of the other systems, other part of the neuroaxis and other system involvement, one important differential diagnosis is mitochondrial disease. So under the myopathy, myopathy means mainly only muscle. Cytopathy means muscle plus other tissues. So myopathy, progressive external ophthalmoplegia, class 1, complex 3. And class 1 means nuclear complex 3 deficiency. Class 2, progressive external ophthalmoplegia, Kearns-Sayre syndrome, deletion tRNA. And this is myopathy, cardiomyopathy, and sporadic cytochrome deficiency. So they are named like this. Mimica means myopathy, cardiomyopathy. Cytopathy means more than muscles, other organs are affected. So heterogeneous, single to multi-organ, different phenotypes with the same mutation or vice versa. The mutation may be same, but clinically there may be different expressions. They present with headache, seizures, Mendel's abnormality, encephalopathy, neuropathy, cranial nerve, 8th and 2nd, and other systems. So mitochondrial muscle disease is the commonest form of metabolic myopathy. The clinical phenotype is remarkably variable and includes purely myopathic disorders as well as multi-system disease in which myopathy occurs in association with symptoms of brain, heart, endocrine and other organ involvement. The muscle and the brain requires a lot of energy. We already saw that the disease becomes more severe in organs which need more ATP. So the brain and the muscle need a lot of ATP, so they become more symptomatic and are therefore often the most severely affected structures. And pathology is impairment of oxidative phosphorylation in the mitochondria and generation of reactive oxygen species. Muscle symptoms are also diverse. It can be just weakness, especially at the extraocular muscles which have maximum mitochondria. And it can be exercise intolerance. After exertion, patient feels unusual fatigue. It can be severe cramps in unusual locations. Air hunger on exertion because the lactic acid accumulates. So after exerting, the lactic acid accumulation results in air hunger. So they will open their mouth and breathe. And myoglobinuria. Exertional fatigue is a cardinal feature. And traditional histochemical, immunohistochemical, and biochemical testing combined with molecular genetic techniques, namely the next generation sequencing, gives the diagnosis. So to suspect when these features are there and then do the various tests and then you can confirm the diagnosis. So fatigue is there. So is it myasthenia? That is a common question asked. So that is neuromuscular junction and both have fatigue. So how do we differentiate? So mitochondrial disease, there is exercise intolerance. After exercise, the patient feels very tired, but there is no drop in the power grade. Whereas in neuromuscular junction, after exercise, drop in the power grade will be demonstrable. Mitochondrial disease patient after exertion, because of lactic acid, they have tendency to breathe through the mouth. So we will think whether the patient is having asthma or heart disease. So that is air hunger. That is not there in neuromuscular junction. Muscle pain, that is not there in neuromuscular junction. Myoglobinuria, not there in neuromuscular junction. Neuromuscular junction is asymmetrical. This is symmetrical. So that is how you differentiate even though both present with fatigue. Then chronic progressive external ophthalmoplegia. The age of onset is below 30 years. Symmetrical, occasionally asymmetrical, non-fatigable. No diplopia. They can have RP. They can have subclinical myopathy. CK can be elevated. EMG, lactate are not helpful in the resting state because the major muscles are not involved. So EMG done in other muscles or lactate assessment will not be useful. Like the diffuse cytopathy. Biopsy of the unaffected muscle will be not giving any clue because the eye muscles we don't generally do biopsy. And electron microscopy done in the affected muscle will show paracrystalline inclusions. And respiratory chain assay will be useful. So this is one of our child. So this is the drooping in mitochondrial disease. You can see the drooping. There's a progressive external ophthalmoplegia syndrome, symmetrical, and they don't have any fatigability. Whereas this is myasthenia. You can see the asymmetry. This is symmetrical. This is asymmetrical. Left is more toast than the right side. And here you see what sign of myasthenia, plus minus sign. When you close one eye, the other eye opens. When you close the other eye, this eye opens. That is called plus minus sign. That will be not there in mitochondrial disease. Then Kearns-Sayre syndrome and NORP, they present with heart disease, ataxia, and elevated CSF protein, and age is before 20 years. They have asymmetrical ptosis. Unlike the progressive external ophthalmoplegia syndrome, Kearns-Sayre syndrome can have asymmetrical ptosis. They can sometimes experience squint diplopia and myopathy. NORP is a neurogenic muscle weakness. So it's a distal weakness with myopathy, ataxia, RP, decreased sensory loss, migraine, dementia, and seizures with a mutation in this locus. So next is a 8-year-old child. So this is the third child. First we saw the lease. Second we saw the progressive external ophthalmoplegia and combined myasthenia. Now this is the third case. 8-year-old child had progressive cognitive decline and diarrhea. So she presented to us at that time when she was 8 years old. And imaging at that time showed white matter changes. So we thought the different possibilities of celiac disease as well as mitochondrial disease. So to rule out celiac disease, we did the small intestinal biopsy, but it did not reveal celiac disease. Therefore, by exclusion, at that time, the patient could not afford genetics, and genetics was not done in the institution where I was working. So they have to send it to the private sector, which was expensive. So by exclusion of celiac disease, we put her on mitochondrial cocktail. Patient became slightly stable, but because she was going on having diarrhea, the family could not believe that a neurological disorder can present with diarrhea. So she was lost for follow-up for several years. And for her GAT symptoms, she was treated from different institutions with various antibiotics with various diagnoses for several years. Slowly, she became deaf. She developed peripheral neuropathy, severe muscle weakness, and progressive dementia. At that time, again, she reported back to us. Then we did the genetics at this time, and it was confirmed as Mingi. I will show the case. And what all the conditions where you get diarrhea with neurological deficit, cerebrotendinous xanthomatosis, mitochondrial gastrointestinal disease, celiac disease, Whipple's disease, malabsorption syndrome, and thyrotoxicosis. So this child was a case of mitochondrial gastrointestinal encephalopathy. If she has been on the cocktail, at least the course could have been slowed down. So this is her second admission. You can see the white matter changes, the ragged red fibers, and you can see the lactate peak, and she has the peripheral neuropathy, and this is the Mingi genetics. So at this time, it was too late, and we lost the patient. This is about 10 years after the initial presentation to the same institution, she came back. Next is the case 4. She is a 13-year-old child who presented with features of juvenile myoclonic epilepsy. Because the drug of choice for juvenile myoclonic epilepsy is sodium valproate, she was treated elsewhere with sodium valproate. Following sodium valproate, she became disoriented, she became blind, and she became deaf. So they were afraid, and the mother all brought her to our hospital, our institution. So why this much complication happened with the anticonvulsant? Then when we saw that the mother and aunt were all having a midline lipoma in the neck. Child did not have the lipoma. Child developed valproate-induced severe toxicity, and the family members showed midline lipoma. So sodium valproate is a mitochondrial poison, and midline lipoma is a systemic clue for mitochondrial disorders. So that confirmed the diagnosis for us. And this we published, and this is the midline lipoma. This is the mother of the child, and many other sisters of the mother also had the midline lipoma. This is in mitochondrial encephalopathy, ragged red fiber syndrome. which present as mental retardation and epilepsy. It is said that if you disturb this like keloid, it will grow very severely. So because this lipoma was becoming very disfiguring for her and she did not have epilepsy, she underwent surgery even though she was told not to do it. And after surgery, this is how it became a terribly expanded lipoma after surgery. So that is mitochondrial encephalopathy, ragged red fiber syndrome. Next is a young boy who was suffering from migraine headache. So migraine can be a feature of mitochondrial disease. It was poorly responsive to treatment, and following an attack of headache, patient presented with seizures. He did not have any deficits, and MRI showed trans-territorial diffusion restriction. That means diffusion restriction not respecting a vascular territory. Here you can see vascular diffusion restriction involving part of anterior cerebral artery and part of middle cerebral artery. So it is a trans-territorial. It is not in the territory of one vessel. And then patchy restrictions in the cerebellum. So that is a typical picture of MELAS, mitochondrial encephalopathy, lactic acidosis, and stroke-like episodes. They present with migraine. Next is a 10-year-old child presented with excessive sleepiness, seizures, poor vision and hearing. She was ataxic, had myoclonus, and she had step-like progression with every infection. And she was hairy. So SURF1 mutation presents with excessive hairiness. She passed away at 12 years following a respiratory infection. And this is the child. Her whole body was covered with hair. Then exertional dyskinesia. Two brothers presented with paroxysmal kinesiogenic dystonia. And as you know, this is channelopathy. Paroxysmal kinesiogenic dystonias are treated with diphenylhydramidine or carbamazepine as they are considered as seizures of the basal ganglia. She was treated like that, but there was no response. As there was no response, the treating doctor put one of the two brothers, both of them had, put one of the child with valproate. And following a single dose of valproate, the child developed severe myoglobinuria, renal shutdown, and died. So the parents got very panicky and they rushed to the teaching institution where I was working. And this history of valproate-induced death, like the valproate-induced encephalopathy in the MERF family with a midline lipoma, this valproate-induced death gave the clue. And they gave this history gave clue for the younger one. And initial all tests and including genetics was negative. During the initial admission, everything was negative, but we put the patient on mitochondrial cocktail, considering the clue from the other sibling. 4 years later, mutations with severe complex 1 deficiency was identified in the same sample. Because in the initial admission, our institution did not have the infrastructure to study oxygen transport chain. But in the same tissue, 4 years later, when the infrastructure improved, so it is to insist on the importance of clinical diagnosis. Because only 4 years later, you could confirm the diagnosis. But the clue was the death in the other child. Otherwise, this child also would have died if you have not missed, if you have missed the diagnosis. So this is also published. And these are another 2 brothers. So this exertional dyskinesia in mitochondrial disease, we described it for the first time in the literature. Same thing with this also. These are mitochondrial disease with the spinal arachnoid cyst. So these 2 brothers presented with progressive ophthalmoplegia and progressive paraplegia. And the paraplegia was due to this arachnoid cyst. A large arachnoid cyst was there. So that was operated and the child could walk much better. You can see the large arachnoid cyst. This association also we have reported for the first time. And there was complex 1 deficiency. And this is 2 sisters having ophthalmoplegia. Uncomplicated ophthalmoplegia, other systems are normal. So next, these are various conditions. We have seen many of the conditions I have shown in this group only. And this is another child who presented with the progressive ophthalmoplegia, learning deficiency, defective hearing, and imbalance. And you can see the progressive external ophthalmoplegia. This is the classical MRI appearance showing symmetrical white matter changes which pair the U fibers. And there is also involvement of the cerebellum. So that is the classical imaging picture of Kearns-Sayre syndrome. And this is another child who developed acquired mitochondrial encephalopathy. This was due to snake bite. So snake venom contains mitochondrial toxin. So child was given antivenin. So came out from the direct effect of the snake bite, but child went into a mitochondrial encephalopathy. This also we published, and this is due to the snake venom induced mitochondrial toxicity, symmetrical caudate, putamen and globus pallidus signal changes. And this was later, after the antivenin treatment was over, treated as mitochondrial encephalopathy and child improved. And this is a 4-year-old child presented with apparent normal early development and had fever and seizures. So naturally, people diagnosed it as encephalitis. I told about the bottleneck effect. So if the number of defective mitochondria is less, the child may be apparently normal. Only during stress, they become symptomatic. So with fever, the oxygen requirement increases and child develops seizures. So people diagnosed encephalitis and treated with acyclovir. Acyclovir is a mitochondrial poison. Like white sodium valproate, acyclovir is a mitochondrial poison. So child rapidly deteriorated and died. So this is not the feature of herpes encephalitis. All of us know that herpes encephalitis produces bilateral symmetrical temporal lobe involvement. Here we are finding caudate as well as putamen and globus pallidus. So this is mitochondrial disease worsened by use of acyclovir. So these are some common cases. All these cases are my own patients. And we can do simple test apart from the genetics and other things. Muscle biopsy, we can do simple test. You can do the modified forearm ischemia test. You have to collect several ml blood in a chilled sodium fluoride treated bottle for lactate and sodium heparin treated bottle for ammonia and evacuated tubes vacutainer. After baseline samples are drawn, a sphygmomanometer cuff is placed around the upper arm, inflated to 30 millimeters of mercury above the systolic blood pressure, and the subject performs a maximum isometric contraction of the forearm flexors using a hand grip dynamometer. So with the BP apparatus. So higher baseline lactate and modest elevation of the lactate after exercise is a clue. It's a very simple test anybody can do. So baseline lactate is high and after exertion, the lactate becomes modestly elevated. That is one of the clue. But during an encephalopathy, even this exertion can produce myoglobinuria. So when the child is acutely sick, we don't do that. But it's a very simple test. And a venous oxygen level. We know that oxygen extraction is not taking place due to the mitochondrial defect. So maximum voluntary contraction using a custom-built dynamometer assessed as the highest of 3 brief maximum hand grip efforts. So subject performs continuous submaximal aerobic hand grip exercise consisting of 5 seconds of isometric grip force alternating with 5 seconds of rest for 6 to 7 minutes. So he will grip, he give a rest, then again give a grip, and this continues for 6 to 7 minutes. For the first 3 minutes of exercise, the target grip force is 30% of maximum voluntary capacity. And for the last 3 minutes, it is about 50% of maximum voluntary capacity. A cardinal feature is increased venous oxygen. So this is one rare condition where the venous oxygen becomes high. Why? Because the oxygen extraction is poor. So venous oxygen remains high. That is another clue. So baseline lactate and modest elevation after exercise. And after exercise, venous oxygen becoming more. That is a second clue. For places which are resource restricted, you suspect the syndrome, and these simple tests can be done anywhere. And then you have got the induced spectrophotometric evaluation of individual respiratory chain complex activities seen only in very few centers, and the fresh tissue should be examined. You cannot do biopsy and transport the tissue in formalin. You cannot do that. So fresh tissue has to be tested. So it should be done where it is available. It is available in the National Institute. Other places I am not aware. Then you have got blue native acrylamide page assay and growth differentiation factor 15 assay and genetic testing, which may not be always positive. So the hyperammonemia syndromes, that is L-arginine related disorders, and also this shows a bulky cortex. So that will resemble mitochondrial disease. The ornithine transcarbamase deficient state, arginosuccinic aciduria, these conditions, it is not the basal ganglia, but it is the bulky cortex. Next is MALOS. I showed one case of migraine-like presentation, non-territorial restriction. These are some other patterns of diffusion restriction, non-territorial in MALOS. And this is also another MALOS, non-territorial diffusion restriction, not respecting an arterial territory. Kearns-Sayre syndrome, I showed the case and the typical MRI. This is another MRI of a typical case, but this is taken from literature. That is my own case. So that is sparing the U fiber. Then you have got Paul G syndromes, the inability to replicate mitochondrial genomes by the mitochondrial DNA polymerase. We saw the homeostatic disorders. So what all types of homeostasis happens in mitochondria? So this comes under that group. So the inability to replicate mitochondrial genome by the mitochondrial DNA polymerase leads to a subset of mitochondrial diseases. They are called Paul G syndromes. So many mutations in the Paul G gene, the gene which encodes the polymerase gamma, have been associated with mitochondrial diseases. They are very severe syndromes: Alpers-Hutton-Locker syndrome, childhood myocerebrohepatopathy spectrum disorders, myoclonic epilepsy, myopathy, sensory ataxia, ataxia, neuropathy spectrum, autosomal recessive progressive external ophthalmoplegia, autosomal dominant progressive external ophthalmoplegia. So these are Paul G syndromes are a group of mitochondrial disorders where the mitochondrial homeostatic mechanisms are defective, and they are very severe syndromes and usually are fatal. So when do we suspect mitochondrial disorder clinically? Clinical scenario, very young onset, worsening in the setting of high metabolic demand like fever or after exertion, and ataxia with or without cerebellar degeneration, and early onset hearing loss, retinal degeneration, optic atrophy, ophthalmoplegia, sudden onset status epilepticus, recurrent physiological triggers like infection, exertion, severe episodes of seizures, good interictal period. In between, they may look normal and worsening with sodium valproate. So suspect mitochondrial disorder when there are multiple sites of neuraxis involvement. Then you have got, we saw the Walker criteria, and we have got so many other criteria. Next is the Bernier criteria. We got the major criteria and minor criteria. All three conditions fulfilled: major, unexplained multi-system disease, progressive course with episodic exacerbation, or family history of mitochondrial DNA mutation, and other metabolic and non-metabolic disorders excluded. You should exclude phenylketonuria, glutaric aciduria, those conditions. And histology will show ragged red fiber. Enzymology will show any of these defects. And functional test fibroblast ATPase synthesis is less than 3 standard deviation below normal. And you will show the genetic abnormality. So that is a major criteria as per Berner criteria, and minor criteria we have got pathology and clinical pictures of compatible respiratory chain disorder and enzymatic abnormality, fibroblast culture, molecular genetics, and metabolic, more than one metabolic indicator of impaired respiratory chain function, that is elevated lactate. So these are minor criteria for Berner's. Then we have got Morava criteria. There are clinical features and CNS features, multi-system features, metabolic and imaging features, and morphological features. So these are varying criteria where different features are clubbed to form the criteria. So the diagnostic workup, simple tests provide important clues. So that is a forearm exercise test, venous oxygen high after exercise, and they may have diabetes, hypothyroidism, serum and CSF lactate may be high, ECG may show conduction block, asymptomatic but abnormal auditory evoked response, and characteristic MRI picture for the various types of disorders which we saw: abnormal nerve conduction, then muscle biopsy, electron microscopy, respiratory enzyme analysis, genetic testing, and each investigation is complementary and not exclusive. Then biochemical test, you do the lactate pyruvate and important marker of mitochondrial disease, that lactate pyruvate. But you have to remember that lactate can be normal. It need not be abnormal always. Only during the acute symptomatic phase, it will be elevated. Other times, it can be normal. Sometimes the blood lactate can be normal. CSF lactate may be elevated. We don't always go and do the CSF. The lactate during episodes of decompensation only and not otherwise. Or spurious lactate elevation when the collection of the blood sample is defective in a struggling child. So lactate can be elevated without mitochondrial disease. It can be normal with mitochondrial disease. That is what you have to remember. And we had the tandem mass spectrometry, elevated alanine, and indicator of chronic pyruvate accumulation. And organic acid, you might find ethyl malonic acid and methyl malonic acid. We saw that in the first child I showed about the least disease. And they can have diabetes, they can have hypothyroidism, they can have other system involvement, including ammonia. Biopsy, ragged red fiber in the modified Gomary's trichrome. Then you have got succinase dehydrogenase, the histochemistry, and you can show which detects the mitochondrial aggregates in the subsarcolemmal region of the fiber due to mitochondrial proliferation. That is accumulation of mitochondrial material which should be eliminated. Mitochondrial homeostasis disorder detected by STA stain. Then presence of cytochrome C oxidase, negative fibers. So cytochrome C oxidase is part of the oxygen transport chain. So COX deficient fibers, demonstrating COX deficient fibers by sequential COX and STH histochemistry. A mosaic pattern is commonly observed with both negative and positive fibers. Negative fibers appear blue and the normal fibers appear brown. And frozen muscle enzyme activity for the various enzymes of the oxygen transport chain. So we can see that this is the ragged red fiber. In the modified Gomary's trichrome stain, this is a ragged blue fiber. This is the NADHDR, this is SDH stain, this is NADHDR stain, ATPase stain, and ATPase at pH 4.6. So each one gives a clue regarding the various components of the mitochondrial chain that is absent. Then importance of stains. So SDH stain, mitochondrial disorder, central core disease, target fiber, moth-eaten and lobulated fibers. So each stain has got used in one type of condition. You can see the moth-eaten fiber in central core disease. And this is the lobulated fibers in the NADH, tubular aggregates in the tubular aggregate myopathy. So that's all. These are not mitochondrial disorders. So treatment is careful nutritional balance. They should not be starving. And fever should not be allowed to persist because fever is metabolically demanding. And avoid strenuous exercise. Avoid starvation. And symptom management for whether it is seizure or cramps or muscle pain or diabetes, treat that. And disease management for heart, diabetes, etc. And mitochondrial cocktail consists of varying substances, but all are not available in our country. Creatine is a reserve for ATP. The dosage in adults is 10 grams per day, split into 2 doses. But this is not easily available. Creatine is not easily available. And in children, it is given 0.1 gram per kg per day, split into 2 doses. Carnitine is available. It helps improve the efficacy of ATP production by helping pull in specific fuel molecules such as fatty acid into the mitochondria. Dose is 20 to 100 milligram per kg per day. Coenzyme Q10 2 to 8 milligram per kg, and it is part of the electron transport chain. And so you can use coenzyme Q10, L-carnitine, and you can use creatine monohydrate, thiamine 100 milligram, alpha lipoic acid 200 milligram, arginine 500 milligram. This becomes a huge amount, but we have to convince the patient these are all oxygen transporters only, so the side effects of drugs will not be there. Then you have got nicotinamide riboside, a form of B3, and ketogenic diet, which consists of low carbohydrate, high lipid content, which helps the lipid utilization by the mitochondria. avoid mitochondrial toxin. That is sodium valproate, phenobarbital, lasers, antiviral agents, anti-HIV retroviral agents, statins. Then elamipritide, mitochondrial intermediate lipid cardiolipin. That is also not available in our country. And vitamin E derivative acts as a potent respiratory reactive oxygen species scavenger. Riboflavin, high dose riboflavin. L-arginine, 500 milligram per kg per dose. This is available. Then thiamine, that is B1, vitamin C, E, alpha are part of the cocktail. Polinic acid, leucovarin, 0.5 to 2.5 milligram per kg. Dichloroacetate for lactate lowering, 10 to 50 milligram per kg if there is high lactate. Hyperammonemia and acidosis is treated on their own merit with ammonia scavenging and alkali. Anesthesia is should be given only with servoflurane when there is complex 1 deficiency. Then these are the various toxins: valproic acid, antiretroviral agents, statins, aspirin, aminoglycosides, and chemotherapeutic agents, acetaminophen, metformin, beta blockers, steroids, and laser. Prevention, genetic counseling of couples, family history analysis, and prenatal diagnosis are important. Males can be told they are less likely to transmit, though Mendelian transmission does occur. Prenatal diagnosis and pre-implantation genetic diagnosis is important. Mitochondrial replacement therapy. That is, you can remove the cytoplasmic mitochondria if it is sick and replace it with a healthy mitochondria from a donor. Or you can have mitochondrial donation in IVF. Oocyte donor. The sperm can be fathers and the egg can be from the donor. So these are various methods which are advocated. And you have got some research materials which are under study. They have not come into practice. Then mitochondrial transplantation, endonuclease targeting the mitochondrial DNA. These are all hypothetical and not into practical treatment. So it can present at any age. Fatal familial lactic acidosis, Leigh syndrome, Alport syndrome, Melas, Menge, Palsy-associated syndromes, external ophthalmic hemiplegia, Melas, NARP, and sudden onset, rapid progression may be seen, and physiological stressors act as triggers. Viral illness, bacterial infection, pregnancy, delivery, surgery, all produce a relapse. So the importance is high degree of suspicion. Investigations can be negative. Keep using clues to save life. Normal lactate muscle biopsy does not exclude mitochondrial disorder. Use all clues. One child we saw the mother having lipoma. Another 2 children we saw valproate toxicity. So that kind of indirect clues. Perform advanced test to establish diagnosis. That is important at some stage, but it's not available to everybody and everywhere. Then molecular genetic confirmation is challenging conclusively, but if it is there, it establishes the diagnosis. It can be negative. That does not exclude the diagnosis. Finally, the phenotype and genotype correlation is not that robust in mitochondrial disorders because of the bottleneck effect, and they are evolving slowly. So here I thank all patients who willingly allowed their pictures for learning purpose. So thank you. Any questions are there?